So the concept was to use, to make these enteroids by just doing endoscopic biopsies. We do endoscopy all the time. You take a tiny biopsy and you're able to create these enteroids. You need very little. Luckily the material is very stable, can last for 24 hours on ice. Or you can take surgical resections that are going to be discarded. And we were able to make these enteroids. And here's what they look like. They start looking like a little spheroid. Over time they start growing. And then they develop into two forms. But this is what a normal piece of intestine would look like. These are the crypts hanging off. Sometimes they form cysts. And this is just a nicer picture of them, loaded with a dye to measure intracellular pH. But this is one of the crypts from the areas that are producing the cells. These cells polarize. They just look like normal intestine. And here in a confocal micrograph you can see there's green and there's red that are separated. This would be the apical membrane of the cell, in which a protein called vilin is present. And the red is a sodium pump, which is only in the basolateral membrane. Blue is the nuclei. And when you differentiate them, I don't think you can see it so well, but that NHE3, that sodium hydrogen exchanger that does absorption, is shown here in green. And the CFTR that does chloride secretion is shown here in red. And normally the CFTR is in the cell waiting to be turned on, and NHE3 is working on the surface. Here's another picture, NHE3 and CFTR, multiple other transporters I won't tell you about. And you can measure sodium hydrogen exchange activity, and it's inhibited by cyclic AMP, which is with the normal regulation. It can be a model of cholera. If you take cholera toxin and expose them, here is normal NHE3 function, and here you can see in the presence of cholera toxin, it's been totally inhibited. And this is a volume measurement. This is another one of those enteroids in which we labeled with two dyes. A red dye is under your fat resting condition, and you can measure the volume. And then after adding cyclic AMP, in green, you can see that the volume markedly increased. And this is chloride secretion occurring through those chloride channels in this little piece of human intestine. This model can take up rotavirus. Rotavirus is one of the leading causes of death in diarrhea throughout the world. And this is a protein that it makes called NSP4, which is an enterotoxin, being taken up into these enteroids. It can be used and this is another picture of those the same enterotoxin. You can use it as a diagnostic test. You can take stool that you don't know if it has rotavirus, expose it to these enteroids, wait a few hours, and then see if it's actually gotten the NSP4 toxin in it. So if you have the toxin in there, only rotavirus makes it. So it's a diagnostic test. And here, rotavirus is inhibiting NHE3. So it's it can be used as a functional unit. Now, that's for the understanding of looking for drug therapy of diarrhea. You can use it for understanding physiology, as we're studying diarrheal diseases, how do diarrheal diseases occur. And you can use that for the high throughput screening that Jin Lu was telling you about. You can then take a bunch of drugs that are either known as effectively released by the FDA, drugs that are already been approved, or you can take unknown drugs. And you can screen, and that's where we are now in this project. We're trying to screen to see if we can find new drugs. But for regenerative medicine, this is when you have too little intestines. So what do you do with that? And this is a platform, a flat platform, that we've placed these enteroids and break them up, and they start forming sheets. And you can make sheets of intestine that can be made into tubes. And based on the characteristics, you might be able to replace the intestine of a child who has too little intestine. Yeah?
Is the difference between an enteroid and just regular cells in a dish that the enteroid is actually functional, whereas the cells in a dish are just sitting there not working together?
Well, the cell in a dish can come from different sources, right? Till now, before this, it was cancer cells in a dish that you had. And those were the only ones that would propagate in culture, in intestine. And these are normal, these are from normal people. When we do endoscopic biopsies, we are looking for the control people to turn out to have nothing wrong other than, they're either screening for colon cancer or really no disease. So this is normal intestine. So this is the concept. So our conclusions are that human duodenal enteroids can be cultured from isolated intact crypts obtained from adult human biopsies and can remain in culture for a long time. We've already gotten to 30 passages, and some people have gotten to 60 passages. Second, the epithelial cells of the human duodenal enteroids are well polarized. Third, the human enteroids express the major intestinal transporters that normally occur there, and they're localized in the right places. And fourthly, these duodenal enteroids are functional, can be used as a model to measure absorption and secretion under both physiologic and disease-mimicking conditions. Human duodenal enteroids can be used as models of cholera and rotavirus. Those are the ones we've studied so far. Human enteroids can grow as monolayers, giving hope that they might be used in regenerative medicine, and they're now being used for antidiarrheal drug development. So thank you.
You've described a marvelously clean system, it seems to me. But everybody, not everybody, but lots of people are really excited about the important role that the microbiota play. Can you incorporate them into this system, or do you just have too much of a mess to deal with?
No, it's a really good question. So actually, I didn't say it, these guys have their own microbiome.
Oh, they do?
We can't get rid of them.
Oh.
So we are now trying to figure out what are the bugs that are there and what's the significance of them. But you're right, that is a pretty hot area, I must say. Yes?
It's just a psychosocial note. I appreciate your talk, and my grandfather's sister died as an infant diarrhea, and my great-grandfather died of typhoid. That seems like a long time ago, but these deaths are so shocking, and they seem so sudden when they occur. And they really do have a lot of resonance down the generations. So even though we are not in the same situation in terms of morbidity and mortality as, I don't know, Guatemala, it's very, I just appreciate your talk. I appreciate your work. And it means a lot to a lot of people. That's all.
Yeah, well, thank you. That's where we're working at, right? I think a lot of people really want to solve this problem for a lot of reasons. And hopefully, now maybe we have the tools.
Does where you take the tissue sample matter? And do you need to get a stem cell in there, stem cells in there too?
Yeah, so it's hard to avoid them. That's the good news. There are different ways you can do this. You can start with IPSs, and they can be made to form intestine, as you heard earlier. But it's a generic intestine. The intestine is on a horizontal basis. The duodenum is different functionally than the jejunum, than the ileum, and the colon. This allows us to do biopsies every place, and it breeds true. The transporters that are present normally in the duodenum stay there, and the ones that are in the ileum aren't there, and the ones that are in the ileum stay. So this is a very nice model where you get, luckily with endoscopy, we're going every place, so no problem.
So basically, to really understand it, you'd have to have multiple samples along the continuum of the intestine.
Correct. Now, one thing you did say, that somebody said, the real tissue is moderately complex, and the neural involvement is important. 50% of diarrhea, the rotavirus and the cholera diarrheas, are neurally mediated, and these enteroids that I'm using don't have nerves, but it's quite easy to grow intestinal nerves, and so now we're trying to put them together and see if we can look at that regulation. Yes.
This is sort of out in left field, but it's because my husband died of carcinoid cancer. Any look at the kind of diarrhea that occurs with these neuroendocrine cancers related to the kind that are occurring out in the field?
Yes. So there are many different diarrheas that have different mechanisms, but they all end up causing diarrhea by affecting NHE3 and CFTR. So if you're aiming for a drug, getting to the end stage is the right approach, even though they start by having many different ways they get to that end stage.
Just to follow up on your comment about the nervous system not being there, does Paregoric work with this model, or do you need the nervous system to be there?
Good question. So, there are at least 4 different types of opiate receptors, and there are mu receptors on the enterocytes. So my guess is that the main function is through the nerves. So both are true. A little bit effect on the epithelial cells themselves, major effect on the nerves.
But, I'm not sure you're answering my question. Does Paragard work in this system? Does it have the soothing effect that it does on people?
Yeah, it'll have an effect. It'll change transport.
It does, okay.
Yes.
Yeah, maybe I missed this, but, are you looking at the lymph system, and how this could relate, how the drain, I don't know enough about the bowel, but, I know that when you resect, for example, if you resect the colon, you could pull out some of those lymph nodes, and in a length about, you probably got what, three dozen lymph nodes, something like
Yeah.
that?
I mean, the lymphocytes is what you're asking about?
The lymph nodes,
Oh, no. So our biopsies are tiny.
okay.
You can't believe how little they are. I mean, less than a millimeter. So we don't get any, if we get towards the lymph nodes, we've done something wrong.